Hi guys, my name is Heather. It's so good to see you guys again. So welcome to Module 4. Today we're going to talk about CRISPR-Cas9. It's an amazing and revolutionary gene editing tool that is widely used in scientific research as well as synthetic biology. If you're ever going to STEM in the future, you're definitely going to be hear about CRISPR-Cas9 or even use it in the future research. So before we start the module, please do the following two things. Open up the syllabus and read the linked articles for Module 4, titled Questions and Answers about CRISPR. It talks a little bit about what CRISPR-Cas9 is, which is tightly related to today's topic. After reading the articles, please also open up the Google Form number 4. It has some questions I would like you guys to answer during this module. So if you have done the both things, then good job, and let's get started. So for today's modules, I will first talk about what CRISPR-Cas9 is. And then I will talk about the history of CRISPR-Cas9, how is it originated, as well as the important components of CRISPR-Cas9, which are the guide RNA, as well as the Cas9 enzyme. At the end, I'll talk a little bit about how CRISPR-Cas9 can be delivered to the cell's genome. So the first question is, what is CRISPR-Cas9? So CRISPR-Cas9 is an abbreviation for cluster regular interspace short palindromic repeats. I know it's a long name, and I'll talk more about how this name is derived in the next slide. But as I said before, it's an amazing gene editing tool. It really allows the scientists to edit gene at a specific location, as well as adding a new functional DNA at a desired location. So as you guys can imagine, this will be very helpful for synthetic biology, as well as anything related with gene editing. So where is CRISPR-Cas9 originated? It's actually a defense mechanism adapted from the bacteria against virus. So as you guys can see in this picture, this is a virus and this is a bacteria cell. So when virus attacks bacteria, it inserts its viral genome into the bacteria cell. The bacteria can actually sniff parts of viral genome and incorporate into the CRISPR region. The name cluster regular interspace short palindromic repeats is because in this CRISPR region, it consists of this palindromic repeats as the black rhombus, as well as part of virus genome shown in this colorful square. So why is this helpful? When the virus attacks again, the bacteria can actually recognize that it's not part of bacteria genome because that virus gene is complementary to part of the virus gene in the CRISPR sequence. Then the bacteria can actually make this molecular scissor that can actually cut the virus genome to disable the gene from the virus. And that protects bacteria from being infected from virus. So when the scientists see this mechanism, they're like, maybe we can really incorporate this into not only cutting the virus gene, but as well as human genome as well. And that's how CRISPR-Cas9 is discovered. So before I go into how CRISPR-Cas9 actually works, I want to introduce the two very important components in CRISPR-Cas9. So the first one is Cas9 enzyme. So that is actually the scissor to cut the target DNA. And that is not active when it's just by itself. The second component is the guide RNA. So guide RNA contains a sequence that are complementary to target DNA. So with the Cas9 enzyme as well as the guide RNA, it forms this complex that's active. So with the Cas enzyme that can cut the genome, as well as the guide RNA that actually guides the Cas9 to tell Cas9 where to cut the genome, this complex is now active and ready to do its job. So how does CRISPR-Cas9 actually work? So after CRISPR-Cas9 complex is formed, it can be introduced into the cell's genome. Cas9 is a molecular scissor, and guide RNA, since it has a complementary region to the target gene, it can bind to the target gene. The Cas9 then cut the target gene and forms this broken genomic DNA. This broken genomic DNA has a deletion of the target gene, and it can either be repaired by cell's own repair mechanism and form a DNA that doesn't have the target gene, or the scientists can introduce another functional DNA into the broken genomic and form this final product that has an original DNA as well as the insertion of a new functional DNA. So in the next slide, I have a video kind of summarizes everything that I've been talking about, but I just thought that having animation might be helpful for understanding the concept.
Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
So as you guys can see here, CRISPR-Cas9 is really helpful in the scientific research and advances. So before I go even further to the module, I would like you guys to answer these two questions in a Google form. So in the first question, I'm very curious that if you are a well-known scientist and have access to fabulous lab and abundant funding, what do you want to research and achieve using CRISPR-Cas9 technique? So here I have shown some of the things that scientists right now are doing using CRISPR-Cas9, but please feel free to use your imagination. The second one is, why do you think CRISPR-Cas9 is helpful in synthetic biology? Now in module 2, you can already have a basic understanding about what synthetic biology is and what I just talked about CRISPR-Cas9 as a genomic editing tool. I'm curious to know, what do you think, how these two techniques can be related to each other? So now pause the video and answer the questions, and after answering it, play the video again, and let's continue the module. So I hope you guys already finishing completing the question, and let's keep going. So the last concept I would like to cover is how is CRISPR-Cas9 can actually be delivered to the cell. So now I talked about how CRISPR-Cas9 works, but we need to have a delivery system to have the CRISPR-Cas9 into the cell's genome in the first place. There are many delivery systems out there in the world, and today I will talk the 3 methods how scientists use to deliver CRISPR-Cas9 to the cell's genome. The first one is lymphofunction. So in the lymphofunction, as you can see here, the scientists can combine the plasmid that encodes for Cas9 and guide RNA with a liposome. Liposome is more just like a vesicle, so it's like a sac with lipids in it. So basically when the scientists combine these two together, it forms a liposome nucleic acid complex. And this complex can then be easily taken by the cell. And the plasmid with Cas9 and guide RNA can be introduced to the cell's genome. The second one is lentiviral transduction. In this mechanism, again, the plasmid encodes for Cas9 and guide RNA can be introduced into a 293 T cells, as well as 2 plasmids that encode for virus. So when these 3 plasmids are introduced into 293 T cells, since it has a plasmid that encodes for virus, it can then form this lentiviral particle that has the Cas9 and guide RNA. This lentivirus can then attack a cell and release the plasmid that has the guide RNA and guide RNA and Cas9, and this plasmid can then be introduced into the cell's genome. The last one is electroporation. So in this one, the science first introduced a short electric pulse into the cell's membrane, which caused forming pores in the cell's membrane. So again, this plasmid that has Cas9 and guide RNA encoding plasmid can then be introduced into the cells through those pores. These cells usually need to be cultured later so that all these pores can be grown back again and have a complete cell membrane. So that kind of concludes about today's module. I hope you guys had some fun learning about CRISPR-Cas9. So I just want to have a brief review of what I just talked about today in the module. So the first thing, CRISPR-Cas9 is a revolutionary gene editing technique. It really allows the scientists to specifically cut the gene at the desired location, as well as adding a new functional DNA into the genome. CRISPR-Cas9 is actually adapted from bacterial defense mechanism, as I showed earlier. And CRISPR-Cas9 consists of two important components, guide RNA and Cas9 enzyme. I also talked about the mechanism of how CRISPR-Cas9 works. And at the end, I talked about 3 CRISPR-Cas9 delivery systems. So lymphoection, lentivirus delivery system, as well as electroporation. So in this module, I have 2 activities that I would like you guys to do. So first activity is that Susan has a sickle cell disease, which is a genetic disorder that is caused by a single DNA letter mutation. As you guys can see here, this is Susan's DNA, and here there's a mutation that causes Susan to have sickle cell disease. So I'm wondering if you could draw the genetic gene editing process using CRISPR-Cas9 that will help Susan to correct the genome from this mutation to a healthy DNA strand that doesn't have that mutation. You can refer to a slide I had before to talk about how CRISPR-Cas9 works. You can start by drawing the guide RNA as well as the Cas9 enzyme and how these two can form a complex and then at the end forming this new healthy DNA strand. The next activity is more like a lighthearted, fun activity. I would like you guys to using the CRISPR-Cas9 knowledge that you have today to explain to me what's the meaning of these 2 memes. If you have any other memes that you think you want to share related with CRISPR or science, please feel free to upload it in the Google form below. Well, thank you so much for listening to today's module. I had so much fun talking to you about CRISPR-Cas9, and I hope you guys had some fun too. Feel free to email us at ufr.igem@gmail.com. We're very happy to receive any feedback, questions from you, because we always love to hear from you. Well, don't forget to complete the Google form. And yeah, I hope you guys had some fun, and I'll see you guys next time. Bye.